Alright, so I wanted to make a quick video about this mitosis board over here. So we're talking about mitosis, which is cell division. And in mitosis, this happens in your body cells, like so the cells of your somatic cells. So not in the egg and sperm. This is a little bit different. This is where we have one cell that makes a copy of itself that's genetically identical. So we've talked about the cell cycle in other areas. So after the cell cycle goes through interphase, right, and it's got G1, the S phase, and the S phase is where the DNA is replicated. Then G2, finally we move into prophase. So stage 1 over here is going to be prophase. And there's a couple other things that I want to talk about in prophase. So remember that this is a cell. You're also going to have to be able to identify some of the parts of the cell. So in the beginning of prophase, the main thing that we notice is that the chromatin in the middle of the which is the DNA inside the nucleus is condensed into chromosomes, right? And so we can see these as these replicated chromosomes here. Remember, they replicated in the S phase. And so it is actually condensed into these dark chromosomes, and you can see them under the microscope. And this is within the nucleus. So the nucleus is going to be this kind of the whole blue area here. This whole thing is going to be the nucleus. And sort of the outer blue layer there is going to be the nuclear membrane. So that's going to be the outer layer. And the nucleus is like the entire thing. This green guy inside is the nucleolus. So the green one here is the nucleolus. Other organelles that are important, this cell does have other organelles, they're just not shown in this model because they're not as important for the process of mitosis, are these guys here. And so in this case, we have two of them because they were finished being replicated in G2. So we have two pairs of paired centrioles, which makes a centrosome. So this guy right here, this is going to be one centriole with two centrioles, and this is another centrosome with two centrioles, and these are paired. And so radiating out around them here, these are called the asters or star asters. We didn't talk too much about those. And these start out near each other because they duplicated, like they replicated, but they are going to eventually move to opposite poles of the cell, as we'll see as we go through mitosis. Another thing here is that the outside of this part right here is the cell membrane or the plasma membrane. So this beginning part is prophase, and we can tell that it's prophase because the chromosomes have condensed, so that's like the start of mitosis, right? Mitosis includes prophase, metaphase, anaphase, and telophase. So the start of prophase is when the chromosomes condense, and so this is early prophase. Now we're going to move on to 2, which is still prophase, but it's a little bit later. At this point, we can see that the centrosomes are moving to opposite poles of the cell, or opposite sides of the cell, and this guy right here, this one is the mitotic spindle. So the mitotic spindle is forming in between these two centrosomes. And so that's the mitotic spindle that's forming. You can see the nuclear envelope is still intact here. So we still have the nuclear envelope, but what is missing? The nucleolus. So the nucleolus is actually broken down. So at this point, the nucleolus is broken down as part of prophase. It's a normal part of prophase. The chromosomes, you can see them a little bit better. They are starting to align, and eventually, as the nuclear envelope breaks down, they'll start to attach to some spindle fibers. So here, in late prophase, or some folks call it prometaphase, this is when the nuclear envelope here is going to start to dissolve or break down. So the nuclear envelope starts to break down, and we can see this whole part here. This is the mitotic spindle. And spindle fibers are going to start attaching to our replicated chromosomes. And our replicated chromosomes are moving around, and eventually they're trying to line up right down the middle. Once they line up in the middle, this is 4, this is going to be metaphase. And so all of these replicated chromosomes are lining up in the middle. We call this the metaphase plate or the equatorial plate. And so right here we have our centrioles. They're going to be on opposite poles of the cell. These guys here that are pink, these are going to be the spindle fibers or spindle fibrils here. And you can see that they're going to attach to this centromere. And so they're attaching to the centromere of the replicated chromosome, and they're going to bring it back and attach it to the centrosome. So I want to take a moment and move over and look at this. This right here is going to be a chromosome. Here we go. Let's go. This is a chromosome. And specifically, this one's going to be a replicated chromosome, right? Because remember, we had DNA was replicated during the S phase of interphase. And so here, this blue part here, let's say, was the original DNA here. And then the green part here might be the new duplicated DNA. And they are attached in the middle at the centromere, and that's going to attach them together. And we call these sister chromatids, right? So it's 2 copies of genetically identical DNA. And so these sister chromatids, these replicated chromosomes, are all going to line up in the middle here at the metaphase plate. And so each of these centrosomes are going to send some spindle fibrils to attach here to the centromere. And this is where these 2 are attached together. So now in anaphase, which is the next phase after metaphase, we have, you can see it's a little bit hard to tell the difference between these 2, but the main thing here is that in metaphase, our centromeres are still sort of attached. They're still in the middle, and our sister chromatids are attached at the centromere. But here, you can see that the sister chromatids are actually being pulled apart by these spindle fibrils. And so we have one sister chromatid going to each side of the cell, so they're no longer attached at that centromere in the middle anymore. So this is early anaphase. Now, as we go to later anaphase, you can see that these sister chromatids are being towed to the other side of the cell. This is still going to be the mitotic spindle in the middle over here. And we have not reformed the nuclear envelope yet. So once the sister chromatids are pulled apart, that starts anaphase, and then anaphase continues until those sister chromatids get into the opposite poles of the cell, right? So we have they arrive at the other side, and then they continue into telophase. And telophase is going to start, like the beginning of telophase is going to be when our nuclear envelope is reformed. And so in telophase, when you see the nuclear envelope again, so this guy right here, the blue area over here, that's going to be telophase. The other things that will happen during telophase are that the nucleolus will reform. So now we have 2 nucleoli, right, 1 in each new cell. And our chromosomes will become, they're going to, the mitotic spindle disassembles, the chromosomes are detached from the spindle fibrils, and eventually, once telophase is ended, then the chromosomes will sort of decompress and they'll become chromatin again. And when that happens, when you can no longer see those condensed chromosomes, that's going to indicate that interphase has happened again. As you can see here in interphase, we have, we can't see the chromosomes anymore, so there's no more condensed chromosomes in there, so it's gone back to chromatin. The nucleolus has reformed, the nuclear membrane has reformed, and each cell has just one centrosome. All right, if you have any questions, let me know. Thanks.